life began in a pond or ocean according to the Primordial Soup Theory. The theory suggests that life was the result of the mixing of a number of ingredients: rain, a jumble of common molecules, warm sunshine, and nighttime cooling. Now experts believe the recipe also relied upon the thickener to help gene-like strands copy themselves in puddles for the first time. The finding adds to a growing body of evidence that suggests life may have evolved with relative ease billions of years ago, and therefore it may have done so elsewhere in the universe too. The earliest form of life is thought to have been based on RNA, a nucleic acid present in all living cells. The theory goes that on the evolutionary timeline, the self-replication of RNA strands long enough to be potential genes roughly mark the doorstep to life. Those long nucleotide chains may have been mixed together in puddles with shorter nucleotide chains, with heat from the sun making long strands detach from their helix structures, giving short ones a chance to match up with them and become their copies. But in water alone, when cooling sets in, the long chains snap back into their helix structures so rapidly that there's no time for the matching process. This is known as strand inhibition. Scientists at the Georgia Institute of Technology set out to solve the mystery of how the precursor of present-day genetic code first duplicated before the existence of enzymes that are indispensable to their process today. Instead of performing experiments in water, Christine He and Isaac Walego added an off-the-shelf viscous solvent or thickener to their soup, which contain real gene strands in order to slow down the movement of long strands. It's a little like making them swim in honey. Martha Grover, a professor who oversaw the study published in the journal Nature Chemistry, explained. Applying that to origin of life chemistry seemed obvious because in prebiotic times there probably were quite a few sticky puddles. PhD student Missy added, in that solution, it gives the short nucleotides, which move faster, time to jump onto the long strand and piece together a duplicate of the long strand. In the experiments, it not only worked but produced an encouraging surprise. The DNA and RNA strands folded onto themselves, forming shapes called hairpins, which helped keep the RNA and DNA available for the pairing process, and the shape may also have allowed more potential for functional activity, like a ribozyme, an enzyme made of RNA that catalyzes biochemical processes. If ribozymes evolved in the same process promoting genetic code reproduction, this may have accelerated the path to first life. Missy explained. The researchers used a naturally occurring gene in their experiment, rather than a neater specifically engineered sequence, to show the important role viscosity have played in the primordial soup and replicate conditions on Earth billions of years ago as closely as possible in a lab. Considering a prebiotic soup, it's probably messy. It's got a lot of impurities, Missy said. When we first started out with more impure nucleotides, it still worked. Maybe the same reaction really could have happened in a messy puddle billions of years ago. While glycolene, the solvent the team used, was probably not present on prebiotic Earth, it's thought other viscous solvents were, and the next step is finding a solvent and enzyme capable of joining the smaller strings to larger ones that would have been present billions of years ago.